In this video, I want to discuss MHC processing. So how are these peptides loaded onto MHC? And I want to start with MHC1. So we said already before, MHC1 presents what is going on inside the cell. So we're talking about presenting cytosolic proteins. We also call them endogenous proteins. And this would be any proteins that are inside the cell. And to make sure our own cells don't get fed up with protein, there are what you can think about like wood choppers around, and that's called a proteasome, that chop off endogenous proteins. So let's draw that in. So let's say that's my endogenous protein. It gets chopped off by this proteasome in little peptides. So a wide variety of little peptides. And now on the endoplasmatic reticulum sits something that is called the TAP transporter. There's actually TAP 1 and TAP 2, but we're just going to call it the TAP transporter. And what this does, it takes these peptides and transports them across the membrane. And who is waiting there in the endoplasmatic reticulum? That's the MHC1. And the MHC1 will be loaded with a peptide and then moves through the ER, through the Golgi, to the surface of the cell. And there it's going to show up proudly presenting what it has found inside the cell. So let's load the MHC with one of these peptides here. And remember, not every peptide will fit. They have to be between 8 and 11 amino acids to fit onto MHC1. So not every peptide will fit, but eventually peptides will fit. And also remember, MHC molecules will be never empty. So you're always going to have a peptide on them. And so let's load this peptide. Let's say this is the lucky guy. It gets loaded onto the MHC1 molecule. And now it moves through the ER, through the Golgi, to the surface of the cell, and is now proudly presenting what it found inside the cell. Let's move over to MHC2 processing. MHC2 shows what's going on outside the cell. So we need to kind of get these proteins first inside the cell to get loaded, and then gonna be on the surface of the cell. So how do we get proteins in? How do we sample the outside? Well, through a process called phagocytosis. So these proteins, and we're going to call them exogenous proteins, as we already discussed before, that's an exogenous protein, get phagocytosed. So they're going to end up in the endosome. That's going to be the endosome. And here they get digested. So we're going to get all kinds of different peptides. The MHC2 is also going to be made in the endoplasmatic reticulum. So let's draw that in here. We're going to zoom in to our MHC2. And now there's a big difference because it's not empty and ready to be loaded, because if it would be empty, it could be loaded with all these peptides here that are pumped into the endoplasmatic reticulum via the TAP. So it has a special blocking molecule inside its peptide growth. And this is called the invariant chain. What's going to happen now with this MHC2 molecule, it's going to move through the Golgi stack and then eventually will be diverted to the endosome. And in the endosome, it gets loaded with peptides, and then it's also going to show up proudly presenting what it found outside the cell. So let's follow the MHC2. So it's going to go through the Golgi stack, but then it's going to be diverted to the endosome, and there it's going to be loaded with a peptide, and then it's going to be expressed on the surface of the cell. And you might wonder, so how is it diverted here, and when does this invariant chain get released? And there is another HLA gene which is called HLA-DM. And this guides the process of diverting it to the endosome and then also releasing the invariant chain. So HLA-DM guides MHC2 and takes care of the release of the invariant chain. And then it eventually will also be showing what it found on the outside of the cell, because remember, that comes from an exogenous protein. This concludes the video on MHC processing and loading.